All right, before we finish up Chapter 8, let's start with a little review here. So in this question, you discover a mutation that causes the production of a truncated, which is a fancy word for shorter, non-functional protein. We call that protein A. What type of mutation has most likely occurred here? This one's a little tricky. Think about what the different types of mutations do to the protein. Go ahead and pause the video. Think about that. Okay, so let's look at the different options. We have missense, nonsense, or silent. So the most likely to cause a non-functional protein that is shorter would be a nonsense mutation. In fact, that's the only one that can make a shorter protein, right? Because that is an early stop codon that makes the protein shorter. A missense might make it non-functional. That's a change in the amino acid sequence, but then again, it might not. It's a little complicated. A silent mutation would not affect the amino acid sequence at all, so it would have no effect. Okay, so the answer is B, a nonsense mutation. Chapter 8.6 is probably my favorite topic ever, biotechnology, using all of these things we've learned about to do things in our lives. Biotechnology is humans using biology as technology, right? We're going to talk about plasmids, restriction enzymes, gel electrophoresis. These are all ways to manipulate and analyze DNA. We're going to talk a lot about the polymerase chain reaction, or PCR, which is a standard thing for testing for microbes. We'll talk about the making of recombinant DNA molecules and how they can be applied to medicine. And we'll talk about DNA sequencing a little bit as well. Okay, so this chapter, I want to pose to you a problem. It's a problem that you've probably had in your life. You want to make a mouse that glows in the dark, right? Something that everybody, I think, wants to do. We're mad scientists today. How do we go about doing this? How do we make this mouse glow in the dark? Well, if you're really clever, you look out at the diversity of biology, and we have some things that do actually glow in the dark. There's a whole group of jellyfish that make a protein that glows green. This is called green fluorescent protein, or GFP. I actually have some E. coli in the lab with this protein in them. This protein has a specific shape, and when UV light hits it, they actually glow this fluorescent green color. So you're real clever and you go, I could take the DNA sequence for this protein and maybe I could stick it into my mouse to make it glow, right? But how do you go about cutting out DNA? Well, if you look at bacteria, there's a whole class of enzymes that we call restriction enzymes or restriction endonucleases. They recognize specific sequences of DNA and cut the DNA. They're like little scissors, right? So you take those restriction enzymes and you take the jellyfish and you cut out the DNA for this protein. There's a whole bunch of different restriction enzymes. They're named after the bacteria they are discovered in. So EcoR1 is from Escherichia coli. Hindi3 is from Haemophilus influenza. BAMH1 is from Bacillus, Amylclovatha. I'm not even going to try to pronounce it today. So we look at bacteria and we find all these different ones here. So you take these and you take the DNA sequence that codes for the green fluorescent protein and you cut it out with your little molecular scissors here using restriction enzymes. Okay, where are you going to put this, right? How are you going to move this around? Well, one technique we can use is using bacterial plasmids. you can cut open the plasmid, paste in your little GFP gene, and use DNA ligase to paste it in. And now you have a bacteria that can carry around your green fluorescent protein. Okay, the bacteria is going to glow green, but we want to get it into a mouse, right? That's the ultimate thing at the end of this. Let's back up and talk about what this is, right? We've taken DNA from one organism, from a jellyfish, and added it to DNA from another organism, a plasmid from a bacteria. Why can't we do this, right? DNA is DNA. Proteins are proteins. It doesn't matter where they came from because all of life is related to one another. It all works pretty much the same. So I can take this jellyfish gene and it will work in bacteria. It would even work in you. That's like the ultimate thing. If I was going to have a superpower, I think I'd probably I'd either make myself be able to sense magnets, which would be super cool, or I'd make myself glow in the dark, right? I'd give myself GFP. That'd be pretty sweet. This recombinant DNA, as it's called, are two pieces of DNA that come from different sources, and they're pasted together. We use plasmids as what we call a vector molecule to carry around our DNA. We can also use viruses to do this, but plasmids are really, really easy because we can just put them into bacteria very, very easily. We get them into bacteria just by zapping them. Okay, so I have my GFP gene in a plasmid. Now the hard part, and we're not even going to touch on how this works, but you need to get the gene into the mouse genome. And usually this has to occur at some sort of egg or embryo stage, and we insert it and it's got to get into the genome. This is actually very complicated, and this is why gene therapy, the ability to correct harmful mutations in humans, is so difficult to do, right? We know what to fix to prevent someone from having a genetic disease. We don't know how to get it in there very well, though. So we're going to assume that this works. We've injected our sequence and it goes into the mouse genome. And then we have a glow-in-the-dark mouse. Okay, you might think this is mad science, but actually this is super critical science. This GFP, this glowing molecule, can tell us a lot of information. So if we attach GFP to a different protein, you just tack it on the end, you can actually see where that protein is. So this GFP technique won some people a Nobel Prize. It seems very crazy, but it's very useful for tracking other proteins. And I've done this before in plants. It's so incredible. It gives you the ability to see proteins. It's fascinating. Another way that recombinant DNA actually saves lives and money is this example of insulin. So diabetics are unable to produce enough insulin to regulate their blood sugar. Insulin is a hormone that helps regulate the processing of blood in the sugar. Type 1 diabetes, that is a genetic condition where there's a mutation in the gene that produces insulin. So children that are born with type 1 diabetes have to live with that for the rest of their life. Type 2 diabetes is an acquired form that has to do with diet and lifestyle things, and people, for whatever reason, we don't fully understand it, their body stops making insulin properly. The ultimate end of this is that diabetics need to inject themselves with insulin to help regulate the processing of their blood sugar. In the past, diabetics had to get their insulin from pigs and cows in slaughterhouses. We would extract pig insulin or cow insulin in any animal that was being slaughtered. So not only was that expensive, but it's not widely available, but also some people are allergic to pig or cow insulin. The proteins, the pig insulin and the cow insulin protein, they're very similar to ours, but they have slight differences that can lead to people who are allergic to them. That's like very bad if you're a diabetic, right? You then cannot use those insulin forms. So it's expensive, it's hard to get around, and limited supply, and it could cause allergies. All right, here's where recombinant DNA comes in to save the day. In the late 80s, people realized that they could take the gene sequence, so the DNA sequence for human insulin, and put it into a plasmid. They could take this plasmid and put it into E. coli. We grow those E. coli in a big what's called bioreactor, and the E. coli make real human insulin, 100% identical to the insulin that you or I make, unless you're diabetic. And then we can take that insulin and harvest it. It's much easier to make, it's much cheaper, it's much safer, it's pure, it's identical in sequence to human insulin. And we're using microbes to make it instead of relying on going to the slaughterhouse to get it from animals. I think this is a perfect example of how recombinant DNA technology is so powerful in medicine. Another awesome technique is polymerase chain reaction. We're going to talk about it here, but we're actually going to do it in the lab, which is super cool. Polymerase chain reaction, if you break down the name, polymerase, remember DNA polymerase makes copies of DNA. Chain reaction, it's a reaction that occurs in a chain. This is a way to amplify, which again is a fancy word for copy, make lots of copies of DNA in a test tube. It relies on 3 steps. So DNA, right, double-stranded DNA is held together by hydrogen bonds. In the first step, we are going to denature or break the hydrogen bonds. And to do that, we're going to add heat. We're going to heat the DNA up to 95 degrees C to break the hydrogen bonds. Okay, that splits open the DNA. Then we're going to cool the DNA down a bit to 55 C, and we're going to add 2 short pieces of DNA called primers. We actually make these, and these are what give us specificity in the area that gets targeted. The DNA primers bind or match to specific sequences in the genome. And then DNA polymerase, the enzyme, comes in and extends off of those primers. Now you can see we started with 1 strand of DNA. We have 2 copies of it. This is just like cell DNA replication, except in a test tube. Now you'll notice we have to heat this up to 95 degrees C. Your enzymes, your human enzymes, cannot survive that high temperature. But what is a microbe we've talked about that can? That's right. Thermus aquaticus, that guy Thomas Brock found it in that weird hot spring in Yellowstone. It's a bacteria that can survive high temperatures. So we've stolen its DNA polymerase and we use it in this reaction. So that's 1 cycle. If we do a second cycle, heat it, cool it, extend, we can do lots and lots of cycles, and after many cycles, we'll have billions of copies of this DNA. So let's look at this broken down. Okay, the first step, we heat the DNA, break the hydrogen bonds. We also have nucleotides in here. Those are A, T's, G's, and C's. Our primers, and of course, DNA polymerase from Thermus aquaticus is in here. So we're going to break open the DNA. Okay, that's our denaturation step. Make sure you know these 3 steps, by the way. The second step is annealing. We cool down to about 55 C and let our primers bind, right? They're going to hydrogen bond to the places they match. So we actually choose these sequences here to match specific regions. Then we heat to 72 to let DNA polymerase extend 5' to 3'. And now we've got copies. So we're going to watch an animation of this that I think illustrates the process very, very nicely.
The polymerase chain reaction, or PCR, is a technique that can produce over 1 million-fold amplification of target DNA within a few hours. PCR requires specific oligonucleotide primers and plenty of nucleotides for DNA polymerization. The DNA polymerase must be heat stable because PCR requires repeated cycles of heating the solution to 95 degrees Celsius. Heat-stable polymerase is obtained from thermophilic bacteria or archaea. In PCR, the reaction mixture is heated to 95 degrees Celsius for 30 seconds to denature or separate the DNA strands. The temperature is then lowered to 55 degrees Celsius for 30 seconds, which allows the oligonucleotide primers to anneal to known DNA sequences flanking the target DNA. The oligonucleotide primers, which are usually 20 to 30 nucleotides in length, are made so that they are complementary to the flanking sequences. The temperature is then brought to 72 degrees Celsius, which is the optimal reaction temperature for the heat-stable DNA polymerase. The polymerase uses the primers as starting points for DNA synthesis, adding one nucleotide at a time to create a complementary strand of DNA. The cycle is repeated, starting with the 95 degrees Celsius heating step. Cooling the solution to 55 degrees Celsius allows the primers to anneal. Heating the solution back to 72 degrees Celsius allows polymerization to proceed. After 2 cycles, 4 double-stranded target DNA sequences exist. After the 3rd cycle, 8 copies exist. After the 4th cycle, 16 copies exist. After 25 cycles, about 30 million copies of the target DNA exist. Because PCR reactions require 25 to 30 heating and cooling cycles, a machine called a thermocycler is used to reproducibly and rapidly deliver these cycles.
All right, so we can make copies of DNA. So what? How does that help us do anything? Well, it helps us in research, right? Because we can make lots of copies of DNA. That makes studying DNA easier. But we can use PCR to identify specific microbes, whether they will amplify or not amplify. So we're going to see how we use PCR to test for the SARS-CoV-2 virus. Okay, so this is our nucleic acid detection or PCR test. This is a test that's going to use primers that match the SARS-CoV-2 virus. If the virus is present in your body, the primers will match and we will have amplification of DNA. We will make many, many copies. We could then see that on a gel, or there's other ways to detect it. If you don't have the virus in you, there's nothing for the primers to match to. So there will be no amplification. You'll have no DNA being made and we get a negative result. The way that this works is we need to start with a specimen swab, and the SARS-CoV-2 virus tends to hang out in the back of the nasal cavity. So that's why that swab's got to go way up into your nose. SARS-CoV-2 is an RNA virus, so there's an extra step that's needed. PCR works on DNA. We have RNA. We have to do an extra step to convert the RNA into DNA. This is done by an enzyme called reverse transcriptase. It's the reverse of transcription. It goes RNA to DNA. Once we have the DNA, we add our primers that will match for the SARS-CoV-2 virus. If a patient is positive, if they had the virus, these primers will bind to the SARS-CoV-2 DNA, and we will have many, many copies that get made at the end. If there was no virus present, the primers have nothing to bind to. So there's no copies that get made and the person is negative. So these specific primers that match the sequence of the virus are really critical to this technique. So again, just to reiterate, if PCR is able to amplify, make copies, you had the virus there, right? The primers were able to match and you were able to make copies. if there was no PCR amplification, there was no virus in you. Now, PCR is super sensitive, so we had a lot of problems with this early on when we were COVID testing. If you do enough PCR cycles, even if there's like one virus molecule in a person, maybe not even enough to make them sick, you can still detect it by PCR. So it took us a little while to figure out what threshold to use to say, okay, yes, there's virus there, but it's such a low level that they're not really going to be infectious. So here's a question. How do you see the DNA, right? I say you make millions of copies, but how do you know that that happened? Well, one way you can do this is gel electrophoresis. So we use a gel, which is an agarose gel. It's not like our Petri dishes, that's agar. Agarose is a much finer thing. It's kind of like Jell-O, but it comes from seaweed. And agarose, we can use it to separate DNA molecules based on their size. The electrophoresis part is because we're using a charge, an electric field, to pull the DNA through. So our gel is here. It's like Jell-O. It's got all these little molecules, and our DNA is going to move through it. DNA is negatively charged, so DNA will be pulled toward the positive end here. So with our pipette, we load our DNA into these little wells here. Then we apply an electric field, and it starts to pull our DNA through. You can see here on this gel what are called little bands of DNA. Those are a large number of DNA molecules of a specific size. Now, think about this. There's a bunch of little molecules in that gel. A small DNA molecule is just going to zip through, right? It's going to bounce off the molecules, but it's real small, so it's going to zip through. Something real large, a huge DNA molecule, it's going to bounce back and forth, right? Think like a pachinko machine with all the little pegs in it. The ball falls down, right? The bigger the ball, the more it's going to bounce off and go slower, slower, slower down, whereas a little ball is going to zip right through. So in an electrophoresis gel, small DNA pieces go much farther in the same time, whereas big DNA pieces don't go as far. So on this end, we have what's called a DNA marker. It's known DNA sizes. So this might be 100 bases, this might be 200, 300, 400, 500. And then we have our samples here, and we're looking at the different sized bands here. So in our COVID example, if there was a band, right, you have the COVID virus in you. But like in this lane here, there's no band. That would be a negative. You don't have the virus in you. That's one way we can see DNA. Another way is what I like to call the Maury-Povich test. The are you the father, right? So we have two parents here. Parent 1, parent 2. There's a sequence of DNA in their genome. Parent 1 has what's called a restriction enzyme site, a place that a restriction enzyme can cut this. This whole region is 10,000 bases or 10 kilobases long. This restriction site means that it gets cut into two parts. One of them is 7,000 bases long, the other is 3,000 bases long. Parent 2 doesn't have the restriction site. Their offspring should be a mix of parent 1 and parent 2, right? Because half of your genome is from one parent and the other half is from the other parent. So they should have one of these and one of these. So, I don't know, let's say parent 1 is claiming that they are not the father of the child, right? So what we would do is we would apply restriction enzymes to a sample of each of their DNA, and then we would run it on a gel. Parent 1 has two bands, one that's 7,000 bases, one that's 3,000 bases. Parent 2 has one band, period, that's 10,000 bases. The offspring, if they are the father, has 10,000, right? Half of their genome has that from parent 2. Then they also have the 7 and 3 because they also have half the genome from that father. So you are the father, parent one, right? This is a simple way that we can do DNA identifications of individuals. Obviously, for real human beings, you have to test more of these. You test many, many different sites for these things. But it does use gel electrophoresis to test these differences in sizes. Another way is just straight up sequencing the DNA, right? DNA sequencing uses a modified form of PCR, but it uses special colored nucleotides and a really thick gel that can separate them out. It's a little complicated what's going on here, but ultimately a laser can go through and read these different colors, and it spits out a DNA sequence for us. This is super useful in identifying pathogens because we can just sequence their DNA and we know what species it is. We know even what strain it might be. Okay, that was my favorite chapter, my favorite section by far. So much cool stuff in there. We have those restriction endonucleases, enzymes that can cut DNA. They cut at palindromes. We use plasmids as vectors to move around that DNA. We can paste it in there with DNA ligase. That makes recombinant DNA from two different sources. You cut it, you paste it in. It's recombinant DNA. You use DNA ligase to tape it all together, basically. The polymerase chain reaction, PCR. It's a clever way to make copies of DNA. You use those two specific primers that match a sequence you've targeted and a heat-resistant polymerase, DNA polymerase, to synthesize new DNA. This all happens in a machine that changes the temperature over and over again. Gel electrophoresis can be used to separate different DNA fragments based on their size. All right, that is it for Chapter 8.